Hello friends. In this module of microbial growth, we will talk about the continuous cultures of bacteria, the synchronous cultures of bacteria, and the classification of microorganisms on the basis of temperature, on the basis of oxygen, and on the basis of pH. The cultures that we have so far discussed in the previous module for the growth of bacterial populations are called batch cultures, in which the bacteria grow in a fixed volume of culture medium. Since the nutrients are not renewed, exponential growth is limited to a few generations. Bacterial cultures can be grown as continuous cultures where they are maintained in a state of exponential growth over long periods of time. In a continuous culture, fresh fermentation media is continuously added to the growth chamber while effluent containing the biomass, products, and unused nutrients are continuously removed. Thus, the volume of the liquid in the growth chamber is maintained constant. The growth rate of the cells in the growth chamber is proportional to the rate of loss of cells through overflow and hence automatically to the rate of inflow of the fresh medium. The basic question is, how does the rate of inflow of fresh medium into the growth chamber determine the growth rate of the culture? Continuous cultures are mainly controlled by two factors. First, concentration of a limiting nutrient and dilution rate. Relationship between growth rate and rate of cell loss can be given as growth rate in growth chamber is equal to rate of cell loss through overflow. Growth rate of bacterial cells can be described as dx upon dt is equal to kx, where x is cell mass per milliliter, t is time, and k is the growth rate constant. Since growth rate is equal to cell loss, rate of cell loss through overflow is equal to dx upon dt depends upon the dilution rate d. D is equal to F upon V. Therefore, dx upon dt is equal to Fx upon V is equal to dx, where F is the flow rate, V is the culture volume, and D is the dilution rate. The dilution rate is the rate at which the medium flows through the growth chamber relative to the volume of the culture vessel. Thus, at steady state, where the growth rate is equal to the rate of cell removal, Kx is equal to dx, hence K is equal to d, and thus growth rate is equal to the dilution rate. The most common device for continuous culture is a chemostat. This can be used to maintain a bacterial population at a constant density, a situation that is in many ways more similar to bacterial growth in natural environments. As can be seen in this figure, in a chemostat, the growth chamber is connected to a reservoir of sterile medium. Once growth is initiated, fresh medium is continuously supplied from the reservoir. The volume of the fluid in the growth chamber is maintained at a constant level by an overflow drain. The rate at which fresh medium enters the growth chamber is equal to the rate at which the spent medium leaves the chamber. As stated above, the chemostat is mainly controlled by concentration of a limiting nutrient and dilution rate. All the nutrients are added in excess in the growth medium except one nutrient which may be a growth factor or a vitamin. This limiting nutrient controls the growth rate of the culture, which is determined by the rate at which new medium enters the growth chamber. The final cell density depends upon the concentration of limiting nutrient. If the dilution rate is very low, some of the cells might die due to lack of nutrients because the limiting nutrient is not being added fast enough to keep up with the cell metabolism. At the other extreme, if the dilution rate is very high, the cells might actually be washed out of the growth chamber because the dilution rate is greater than the maximum growth rate. Hence, a chemostat operates best at low dilution rates. In the previous module, we have learnt that when a bacterial culture is grown in a liquid media, the media becomes turbid. The more the number of cells, the more is the turbidity. This turbidity can be measured by means of a colorimeter or a spectrophotometer. Both these devices measure the amount of unscattered light that emerges. Another device for maintaining continuous cultures is a turbidostat. As shown in the figure, it has a photocell to measure turbidity in the growth chamber. Like the chemostat, it has a growth chamber which is attached to a reservoir of sterile medium, and it also has an outlet for removal of the spent medium. The flow rate of the fresh medium into the growth chamber is adjusted in such a way that constant turbidity or cell density is maintained in the growth chamber. Unlike the chemostat, the culture medium does not have a limiting nutrient and also does not have a constant dilution rate. The turbidostat operates best at high dilution rates. Let us now move on to the next type of cultures, which are called synchronous cultures. Studying the growth of bacterial populations in batch or continuous cultures does not allow us to understand about the growth behavior of individual cells because the distribution of cell size among the members of the population is completely random. Not all cells grow and divide at the same time. Information about the growth behavior of individual bacterium can, however, be obtained by the study of synchronous cultures. Synchronous cultures are cultures composed of cells which are all at the same stage of the bacterial cell cycle. Hence, all the cells will divide at the same time, grow for a generation time, and again divide at the same time. Measurements made on synchronized cultures are equivalent to measurements made on individual cells. The synchronous cultures therefore show a biphasic growth pattern. There are two methods available for producing the synchronous cultures. The first are induction methods. They rely on synchronizing an exponentially growing culture by appropriate and usually sudden changes in the environment such as alteration in the temperature, concentration of nutrients or illumination for photoautotrophs. For example, if the temperature of an exponentially growing culture which has an optimum growth temperature of 37 degrees is suddenly brought down to 20 degrees and maintained for some time, the low temperature retards cell division. The low temperature slows down the metabolism and all the cells slowly reach maturity, but none of them divides. Now, when the temperature is again suddenly raised to 37 degrees, all the cells divide synchronously. After growing synchronously for a few generations, the cultures rapidly lose synchrony because not all cells in the population divide at exactly the same time, size, or age. The second methods are selection methods. In this method, from an actively growing culture, the cells at a particular point in the growth cycle are selected and physically separated, say when the cells have just completed cell division. Therefore, the smallest cells in the bacterial population are those that have just completed the process of cell division. The methods used for separation include centrifugation on a density gradient. All the cells of the same size can be obtained, and now when they are grown in a culture medium, they all start their life cycle from the same point. Second, filtration of cells through a cellulose nitrate filter. The most widely used method for obtaining synchronous cultures is the Hemstetter-Cummings apparatus. As can be seen in the figure, it consists of a glass vessel with an inlet and an outlet and a membrane filter with a specific pore size. The pore size of the filter is selected in such a way that the smallest cells will pass off through the filter. These are the cells which have just divided and now would go through the entire life cycle before dividing again. On the other hand, the cells which get stuck up in the filter are the ones which are the largest and which are on the verge of cell division. These can be collected by inverting the filter and passing medium through the filter from above. When the cells divide, they fall off the filter, giving a continuous supply of newly born cells which can grow synchronously. Let us now move on to the effect of the environmental factors on growth. The growth of organisms greatly depend on environmental conditions which are responsible for either accelerating it or retarding it. If we understand these factors, we can understand the distribution of microbes in nature and also devise methods to curb the growth of the undesirable microbes. The environmental parameters that influence the growth rate the most are temperature, oxygen availability, hydrogen ion concentration, and solute concentration. Let us consider each of these factors in detail. Every microorganism has a defined range of temperature within which it can grow. Amazingly, bacteria have been found to grow at almost any temperature where water is liquid. Each organism typically has a range of about 20 to 30 degrees where it can grow, although this can vary greatly. Neisseria gonorrhoeae, a sexually transmitted obligate pathogen, can only grow in a narrow temperature range near 37. In contrast, Bacillus licheniformis is capable of growth from 25 degrees to 60 degrees centigrade. These ranges clearly reflect the environment where the microbes live. Neisseria gonorrhoeae will always encounter temperatures near 37 in its environment, the human body. While Bacillus licheniformis is a soil-dwelling microbe and is subjected to greater temperature fluctuations. Having a wide growth temperature range enables this microbe to take advantage of its habitat. Within the temperature range, there are the cardinal temperatures. These are minimum temperature. This is the temperature below which the growth does not occur. The second is the optimum temperature, the temperature at which the growth is the fastest, and the maximum temperature, the temperature above which growth is not possible. The maximum temperature is rarely its optimum temperature, and the optimum is always almost a few degrees cooler. Classification of microorganisms on the basis of temperature is what we will be discussing now. On the basis of their temperature optimum, microorganisms can be broadly classified into four categories. These are psychrophiles, mesophiles, thermophiles, and hyperthermophiles. Let us understand each of these terms with the help of this figure. Psychrophiles have a temperature optimum below 20 degrees and cannot grow above this temperature. Mesophiles are organisms with an optima in the 20 to 45 degrees range, and thermophiles have an optima from 45 to 80 degrees. Finally, those with the temperature optima above 80 degrees are called extreme thermophiles or hyperthermophiles. Some organisms that normally have their optimum growth rate above 20 degrees can also grow at low temperatures, though slowly. These are called psychrotrophs or psychrotolerants. Listeria monocytogenes, a food-borne pathogen, belongs to this category. This microbe is unusually hardy and will survive many food processing treatments. Ready to serve products can sometimes be contaminated and the microbe will grow to high populations at 4 degree if given enough time. How do these organisms survive at freezing and high temperatures? Obviously, they undergo some adaptations. Let us first discuss the adaptations of psychrophiles. At low temperatures, the membrane begins to solidify, making it difficult for the organism to carry out its many necessary functions. Psychrophilic bacteria compensate for low temperatures by synthesizing lipids containing fatty acids with double bonds. Lipids with unsaturated fatty acids have lower freezing points and thus the membrane will stay fluid at low temperatures, just as vegetable oil made of polyunsaturated fatty acids is liquid even at the refrigerator temperatures, while butter made of saturated fatty acids is solid. Low temperatures also slow the rates of enzymatic reactions, and at some critical level the reactions become so slow that the growth cannot continue. Psychrophiles again compensate for this by changing the structure of their enzymes such that they have more of alpha helix than of beta pleated sheath structure in their proteins. Beta sheet has a tendency to form a rigid structure and hence more the alpha helix content, more is the flexibility at lower temperatures. Also, these enzymes have more polar and less hydrophobic amino acids which might also keep the proteins flexible. Coming to the adaptations of thermophiles and hyperthermophiles, at the higher temperatures, the thermophiles have to deal with a membrane that is too fluid. This great stress might allow the leakage of cytoplasmic contents into the environment. Under these conditions, more long chain fatty acids lacking double bonds are used in the synthesis of membrane lipids. Thus, saturated fatty acids increase the strength of the hydrophobic interactions in the membrane and helps hold it together. Thermal denaturation of protein is also a problem and protein modifications are evident in microbes that live at high temperatures. Their proteins form more compact structures. Some thermophilic enzymes contain greater numbers of salt bridges and it is thought that these ionic interactions play an important role in stability. Comparison of homologous proteins of mesophiles and thermophiles reveal that proteins from thermophiles have more hydrophobic amino acids with branched side chains. These may contribute to a more stable hydrophobic core from thermophilic protein. In addition, loops of the peptide chain that are at the surface and project into the surrounding environment are deleted in thermophiles when compared to their mesophilic homologs. Removal of these surface loops may prevent an active hot solution from tearing the protein apart. Let us now discuss the classification of microorganisms on the basis of the concentration of hydrogen ion, that is pH. It is possible to classify bacteria based upon the pH at which they grow. On the basis of their pH optimum, bacteria are divided into three classes. These are acidophiles, neutrophiles, and alkalophiles. Let us understand these terms with the help of this picture. Neutrophiles prefer a pH in the range of 5 to 8 and the most organisms fall into this particular category. Pathogens of mammals usually have extraordinarily narrow pH ranges very close to 7 where they will grow well. Acidophiles have a pH optima in the range of 2 to 5.5 and include many fungi and some obligate acidophilic bacteria such as Thiobacillus, Sulfolobus and Thermoplasma. The most acidophilic bacteria known is Picrophilus oshime, which grows at pH 0.7 and above pH 4 the cells are lysed. Alkalophiles prefer a pH in the range of 8 to 11 and are found in highly basic soda lakes and high carbonate soils. Surprisingly, acidophiles and alkalophiles maintain their cytoplasmic pH around 7 irrespective of their environment. Thus, optimal growth pH refers to the extracellular pH. It seems they solve the problem of pH extremes by constantly pumping ions in and out of the cell to keep their internal pH neutral. At a molecular level, this makes sense. If the pH of the cytoplasm were allowed to be acidic or alkaline, it would cause the denaturation of enzymes and nucleic acids. Major changes would need to be made to every enzyme and to the structure of DNA for it to function in a very alkaline or acidic cytoplasm. It seems much easier for the cell to spend energy to pump the offending ions out. This does bring up the problem of creating a proton motive force for the purposes of energy generation. For acidophiles, this is clearly not a problem since there is an abundance of protons on the outside. However, for alkalophiles, which grow only at pH levels of 8 to 11, the concentration of protons inside the cell is actually higher. These alkalophiles can maintain a cytoplasmic pH of 9 or lower even at an external pH of 11, thus growing optimally under conditions in which the cytoplasm is more acidic than the external medium. To drive ATP synthesis, it appears alkalophiles have their respiratory enzymes near the ATP synthase such that in pumping protons they create a high local concentration of protons. This is then enough to drive the ATP synthase. Let us now discuss the classification of microorganisms on the basis of oxygen. Microorganisms differ in their requirement of oxygen for growth. At one hand, some cannot survive without oxygen, and at the other, some cannot tolerate oxygen. On the basis of their oxygen requirements, microbes can be classified into mainly five categories: obligate aerobes, obligate anaerobes, facultative anaerobes, aerotolerant anaerobes, and microaerophiles. Let us first discuss obligate aerobes. Organisms that require oxygen for their growth and are capable of using oxygen as a terminal electron acceptor. They can tolerate a level of oxygen equivalent to or higher than that present in an air atmosphere, that is 21% oxygen, and cannot survive in its absence. They have a strictly respiratory type of metabolism and are killed in the absence of oxygen. Examples are Bacillus subtilis, Mycobacterium tuberculosis, Mycobacterium leprae, and Neisseria gonorrhoeae. The next class is obligate anaerobes. These are organisms those are incapable of oxygen dependent growth and cannot grow in the presence of oxygen concentration equivalent to that present in air atmosphere, that is 21% oxygen. Some anaerobes may have a fermentative type of metabolism. Others may carry out anaerobic respiration in which terminal electron acceptor other than oxygen is used. Examples are Bacteroidetes vanilli, Clostridium pasturanium, and Methanococcus. The third class is that of facultative anaerobes. These are organisms that can grow well both in the absence of oxygen and in the presence of a level of oxygen equivalent to that of air atmosphere concentration. Examples are Corynebacterium diphtheria, Listeria monocytogenes, Staphylococcus aureus, Vibrio cholerae, and Escherichia coli. The fourth class is that of aerotolerants. These are organisms that simply ignore oxygen and grow well whether it is present or not. When oxygen is present, however, it is not used as the terminal electron acceptor, and these organisms grow strictly using fermentative metabolism. Examples are Enterococcus faecalis, Streptococcus mutans, and Clostridium tertium. The last class is that of microaerophiles. These are organisms that are capable of oxygen dependent growth but cannot grow in the presence of a level of oxygen equivalent to that present in an air atmosphere that is 21% oxygen. Oxygen dependent growth occurs only at low oxygen levels. In addition to being able to respire with oxygen, some microaerophiles may be capable of respiring anaerobically with electron acceptors other than oxygen. Examples are Campylobacter jejuni and Helicobacter pylori. It is easy to determine the oxygen requirements of a microbe by simple laboratory procedures, one of the easiest being the thioglycolate test, which can differentiate bacteria into these 5 groups based upon their relationship to oxygen. Thioglycolate agar contains thioglycolic acid, cysteine, and agar that restrict oxygen to the top one-third of the tube. After inoculation and incubation, the pattern of growth in the test tube will determine the oxygen relationship. As shown in this figure, obligate aerobes will grow only at the top of the tube where high concentrations of oxygen are present. Facultative anaerobes will grow throughout the tube, but more growth is seen near the top with more oxygen. Strict anaerobes will only grow at the bottom of the test tube where there is no oxygen. Aerotolerants will grow throughout the test tube and microaerophiles will grow below the top layer where low concentration of oxygen has diffused. All cells contain enzymes that can be damaged by reacting with oxygen, and any organism that wants to live in its presence must have methods to protect it. Oxygen will react in the cell to form two major toxic products, superoxide and hydrogen peroxide. It is these molecules that can potentially wreak havoc on a cell by causing uncontrolled oxidations of cellular components. Superoxide is detoxified by superoxide dismutase, which combines two superoxide molecules forming hydrogen peroxide and oxygen gas, and is present in all organisms capable of surviving in air. Hydrogen peroxide is dealt with by the enzyme catalase, which takes two hydrogen peroxides and forms one molecule of water and one molecule of oxygen. Aerotolerant anaerobes have peroxidase instead of catalase, reducing hydrogen peroxide with NADH plus H plus and producing water. Strict anaerobes are unable to survive in the presence of oxygen because they lack these enzymes capable of detoxifying superoxide and hydrogen peroxide. These chemicals accumulate rapidly and kill the cell. Thus, organisms such as Clostridium and methanogens that do not possess these protective mechanisms cannot survive in the presence of air and are restricted to anaerobic habitats. Let us now move on to the methods of cultivation of anaerobes. The fact that aerobes require oxygen to grow and anaerobes are actually killed by oxygen requires vastly different cultivation techniques for both of them. While growing aerobes, it is essential to ensure that they are continuously supplied with fresh air. Liquid cultures are kept in a shaker incubator to aerate them. On the other hand, while growing anaerobes, one has to ensure that there is no oxygen. The methods for cultivation of anaerobes are described now. The first is exclusion of air from the medium. The medium is boiled to drive off dissolved oxygen from the medium, cooled and inoculated, and the mouth of the test tube is sealed to prevent entry of air. Anaerobes grow at the bottom of the tube. The second method is addition of reducing agents. The most common method for growing strict anaerobes is addition of reducing agents like thioglycolate, cysteine, ascorbic acid, etc. to the medium, which brings down the redox potential minus 300 MV or less. A redox indicator such as risazurin, which is pink in the presence of oxygen and colorless in its absence, can be added to indicate the absence of oxygen. The next method is the pyrogallic acid method. This is a routinely used method for cultivation of anaerobes. As can be seen in this figure, the organism to be grown is streaked on the slant and the cotton plug is cut and pushed in with the help of a glass rod. Pyrogallic acid crystals are sprinkled over the cotton and 4% NaOH is trickled down along the walls of the test tube to wet the pyrogallic acid crystals. The chemical reaction between pyrogallic acid and NaOH creates an anaerobic environment inside the tube. The mouth of the test tube is then closed with a rubber stopper and the test tube is kept for incubation in an inverted position. The next method is that of candle jars. We all know that a burning candle placed in a closed jar extinguishes after some time due to depletion of oxygen. The same principle is employed to create anaerobic conditions in a closed jar. Plates and tubes inoculated with the anaerobic organism are placed in the jar with a lighted candle and the jar is sealed. The oxygen is consumed and the carbon dioxide percentage is approximately 3%. Candle jars are particularly used to cultivate capnophiles which require an increased carbon dioxide concentration. The next method is that of anaerobic gas jars. Anaerobic jars or the gas pack system are used to incubate plates in an anaerobic atmosphere. The basic principle of all anaerobic jars is the same. Removal of oxygen from the chamber by reaction with hydrogen added to the system in the presence of a catalyst. The oxygen combines with hydrogen gas and is reduced to water. The anaerobic gas jar is a cylindrical vessel made of metal or glass with an airtight lid. As can be seen in the figure, palladium pellets which act as catalyst are placed on the underside of the lid. A packet containing sodium bicarbonate and sodium borohydride is placed in the chamber which reacts with the moisture to produce hydrogen and carbon dioxide. Oxygen present in the air reacts with hydrogen and is reduced to water. This reaction is catalyzed by palladium. Thus, the air inside the chamber is devoid of oxygen and becomes anaerobic. This is indicated by indicator strips which change the color when the conditions inside the chamber become anaerobic.